All right, now we're going to move on to the different organelles of the eukaryotic cells. So after this section, you should be able to describe the structures of the nucleus and explain its role in the storage of genetic information. You should be able to summarize the functions of the organelles of the endomembrane system and explain the role and location of the ribosomes. Here we go. Oh, there we go. As I sort of mentioned, there are going to be two classes of organelles. There's the endomembrane system, so those will be the organelles that communicate with one another. They're going to do this via membrane channels and small vesicles. The nucleus is going to contain the genetic instructions for the manufacturing of proteins that are involved in most of the cellular functions, and then the endomembrane system is going to be a series of membranous organelles that function to process the materials for the cell. So the nucleus stores that information, and then the rest of the endomembrane system processes that information and puts it to good use. Sort of separately from that, we also have the energy-related organelles. The energy organelles are independent and self-sufficient. They don't rely on information from the nucleus. These will be the structures like the mitochondria and the chloroplasts, who are ultimately going to be responsible for cellular respiration and photosynthesis. So, like I said, the nucleus is going to be responsible for containing the genetic instructions for manufacturing proteins that are involved in most cellular functions. The endomembrane system will be a series of membranous organelles that function to process the materials for that cell. So here we have just sort of an overview of that nucleus and the endomembrane system, or a portion of it. So here we have our nucleus, we have our nucleolus, and then we have the endoplasmic reticulum that's found around it. The nucleus is the command center of the cell. It's usually going to be located somewhere near the center of the cell, and it's separated from the cytoplasm by what we call a nuclear envelope. The nuclear envelope is a double layer of membrane. Inside of that nuclear envelope, we have what we refer to as nuclear pores. Nuclear pores permit the exchange between the nucleoplasm and the cytoplasm, so the material that's inside the nucleus and the material outside of the nucleus. The nucleus itself contains chromatin in a semi-fluid nucleoplasm. That chromatin contains nucleic acids and proteins that will condense down to form chromosomes during cellular division. The chromosomes are going to be our carriers of our genetic information. Inside of the nucleus, we have the dark region right here that's referred to as the nucleolus. The nucleolus is composed of ribosomal RNA, which produces subunits of ribosomes. So here is our nucleus. Our nucleus contains our DNA in the form of chromatin most of the time, and it condenses down into those chromosomes when we are dividing the cell. The DNA is going to be made up of genes which contain the instructions for how to produce proteins. The nucleoplasm is going to be the fluid inside of the nucleus. The nucleolus is that region right here inside the nucleus. This is where we're going to be producing our ribosomes, which we'll get to in just a moment. The nuclear envelope, like I said, is a double membrane around the nucleus that's embedded with nuclear pores that allow for the exchange of genetic material. You can kind of think of your DNA as sort of a hard copy. It's got a hard copy of all of our information. We don't want to ruin it. We don't want to destroy it. We want to protect it at all costs. So it's kind of like your textbook. You put your textbook inside of, I don't know, a book cover to keep it safe. Now, you may scribble all over your textbook, tear pages out. I don't know what you do. That's your textbook. Me in particular, I'm very particular about my textbooks because I always like to sell them at the end of the semester. So I always kept my textbook in excellent condition, safe and secure. But what I would do is go to my textbook and copy down the relevant information in the form of notes. That's what we're going to see with the production of DNA. Or with the processing of DNA. We're going to see that the hard copy DNA is kept safe and sound inside the nucleus, and that we're going to use RNA to copy down that genetic information in the nucleus, and then that RNA is going to leave the nucleus and be processed into DNA, or processed into proteins. And that processing, that synthesis of proteins, is going to occur in the ribosomes. So the ribosomes are our next structure of the eukaryotic cells. They are often called protein factories of the cell. They're going to be composed of a large subunit and a small subunit, both of which are made inside of the nucleolus. The number of ribosomes in the cell is going to vary depending on its function. So for example, pancreatic cells, who are responsible for producing insulin, which is a protein hormone, are going to have a lot of ribosomes. They need to produce a lot of they have a lot of protein secretions like insulin. Ribosomes can be located in one of several locations. They may be located on endoplasmic reticulum. If they are, we refer to it as rough endoplasmic reticulum. It sort of gives it a very bumpy appearance, which you'll see in the next few slides. Or they could be found free-floating in the cytoplasm, either singly or in little groups called polyribosomes. They're ultimately going to be the site of protein synthesis. In the process of transcription and translation, information for the gene is going to be copied down as mRNA. That mRNA can then leave the nucleus through those nuclear pores and travel into the cytoplasm. Once in the cytoplasm, they're going to bind to those ribosomes. That ribosome is going to take that coded message from the DNA and match it up and translate it into the correct sequence of amino acids in order to make a protein. Those proteins that are synthesized by cytoplasmic reticulum or by the cytoplasmic ribosomes will stay in the cytoplasm. Those that are attached to the ribosomes, those by attached ribosomes are going to end up in the endoplasmic reticulum, which we'll get to in a moment. But what we're going to be, the central dogma of molecular biology is DNA goes to mRNA, goes to the protein sequence. And we'll spend a lot of time going over how we can synthesize proteins from DNA, but that's just to kind of give you an overview of where and how this is happening. So next up is the endomembrane system. The endomembrane system compartmentalizes the cell so that particular enzymatic reactions are restricted to specific regions, and overall efficiency is going to be highly increased. So this is going to contain the nuclear envelope, membranes of the endoplasmic reticulum, the Golgi apparatus, the vesicles, and there'll be several different types of vesicles. The vesicles are going to be responsible for transporting materials between the organelles of the system. So here's an overview of the system here. We have our nucleus, our rough endoplasmic reticulum, our smooth endoplasmic reticulum. We're going to have vesicles that are going to transport lipids from the smooth endoplasmic reticulum to the Golgi apparatus. From the Golgi apparatus, vesicles are going to be released and transported to the surface of the cell membrane. We're also going to see vesicles going from the rough endoplasmic reticulum, transported and processed through the Golgi apparatus, and then released in secretory vesicles to the outside of the cell. So we'll go through each one of those. First, we have the rough ER. The rough ER is going to be studied with ribosomes on the cytoplasmic side. This is going to be the site of protein production because, like we said, ribosomes are little protein factories. Therefore, the endoplasmic reticulum that is covered in ribosomes is going to be responsible for synthesizing proteins. Oh, I'm sorry, I have the hiccups. So the rough ER is going to be responsible for modifying and processing proteins. It's going to add sugar to proteins to result in glycoproteins, which are essential to cell functions. They're also going to form transport vesicles that will process and move that cargo to the Golgi apparatus. The smooth ER is called the smooth ER because it does not contain ribosomes. This is going to be, because it doesn't contain ribosomes, it's not going to be responsible for protein synthesis. Instead, it's going to be responsible for the synthesis of lipids. So for example, in testes, the testosterone is going to be produced by the smooth ER. So this will be the site of lipid hormone production. This is also the site of various synthetic processes, detoxification, and storage. So the liver, which is completely abundant with smooth ER, is going to be responsible for detoxifying drugs out of the system. The Golgi apparatus was named for the individual who discovered it, Camillo Golgi. This consists of flattened, curved saccules. It kind of resembles a stack of hollow pancakes. This is going to be responsible for modifying proteins and lipids with signal sequences. So basically, you can kind of think of this as the UPS of the cell, or the packaging center of the cell. UPS does not make product, it only ships it. And so that's going to be the Golgi apparatus. It does not make proteins, it does not make lipids, but it does tag it and then ship it to wherever it needs to go. So it'll receive vesicles from the ER, from the endoplasmic reticulum, and then after modification, it'll prepare those vesicles for shipment. So it'll package proteins and lipids in vesicles that leave the Golgi apparatus to the outer side of the cell. Some are going to be transported to various locations in the cell. Some are going to be exported from the cell through secretion and exocytosis. Some are actually going to be returned to the endoplasmic reticulum or merged with the plasma membrane to help maintain that plasma membrane. So ultimately, the Golgi apparatus is going to be responsible for forming those transport vesicles. Vesicles are going to be small membranous sacs that can be used for transport. Lysosomes are very highly specified vesicles made by the Golgi apparatus that contain hydrolytic enzymes. These are going to be responsible for helping with the digestion and degradation of the cell's waste material. They're highly, highly acidic, which means they have what? Either a high pH or a low pH. I'm going to leave that question up for you. So you determine if we have highly acidic environment, does that mean we have a high pH or a very low pH? They're also going to contain digestive enzymes. Those are going to be used to digest nutrients that are taken into the cell, but they can also be used to break down and remove damaged or old organelles that are already located inside the cell. So this is sort of our garbage disposal. We'll break down materials from the outside, but also old and damaged materials from the inside. In white blood cells, they are going to be used to engulf and destroy pathogens. So this next slide, I think this slide, you've seen it already, is a fantastic summary slide. I would love for you to be able to look at this slide and describe what is happening. Describe what is happening inside of the nucleus. Describe how that information from the nucleus leaves the nucleus and it goes into the cytoplasm, where it's either taken up by the rough ER or free-floating ribosomes. From there, we're going to synthesize proteins, which are going to be secreted from the rough endoplasmic reticulum into vesicles. Those vesicles are going to be transported to the Golgi apparatus, where they'll be processed and tagged and prepared for shipment and secretion out of the cell through secretory vesicles. So if you can kind of talk through everything that is sort of happening in this slide, you'll have a really good understanding of what I would like you to know from this section. So just to summarize, make sure you can describe the functions of the various organelles, endoplasmic reticulum, Golgi apparatus, and lysosomes. Explain how the nucleus, ribosomes, and rough ER contribute to protein synthesis. And then describe the organelles of the endomembrane system that are involved in the export of a protein from the cell. You should also be able to do the same thing for lipids, for the synthesis and the export of lipids from the cell. All right, and that's all I have for this section.